Welcome to Chapter 20, Antimicrobial Drugs, Part 1. This chapter fulfills objective number 4, once again, and the topics will include the interactions between microbes and their hosts, microbes in human disease, and also applied microbiology in this situation, antimicrobials. In Part 1, we're going to talk about the history of antimicrobials, then we're going to talk about the spectrum of antimicrobial activity, the action of antimicrobial drugs, and commonly used antimicrobial drugs. In Part 2, we'll talk about tests to guide chemotherapy, resistant to antimicrobial drugs, antibiotic safety, effects of combination of drugs, and the future of chemotherapeutic agents. In the early 20th century, Paul Ehrlich in Germany made a study of trying to stain bacteria without staining the surrounding tissue in histological specimens. While he was doing this, and when he became successful, it occurred to him that we might be able to do the same thing with drugs or something else that might kill bacteria without harming the surrounding tissue. And Paul Ehrlich referred to this as a magic bullet. He also coined the term chemotherapy. We continue to use that term, but now most of the time when you hear the term chemotherapy, we have a tendency to think of anti-cancer drugs. But it comes down to the same thing. You're trying to kill one type of cell without harming other types of cells. Later in England, Alexander Fleming made a discovery. He found on a plate that he was trying to grow, Staphylococcus aureus, that a contaminating mold colony inhibited the growth of the Staph aureus. Now you've seen inhibition in lab. Remember when we did the Kirby-Bauer test when we put the disc soaked with antibiotics onto the lawn of bacteria and the bacteria grew all the way around the discs, leaving a zone where nothing grew around the disc? Well, Alexander Fleming saw this around a mold colony, and the genus of the mold was Penicillium, so the chemical that the Penicillium mold was releasing became known as penicillin. Now you're familiar with penicillin. We'll talk more about penicillin as we go along. But penicillin was one of the first of these magic bullets, these chemotherapeutic agents that became used in the fight against bacterial infections. Now, penicillin didn't become commercially available until after another chemotherapeutic agent. In Germany, inspired by Dr. Ehrlich, the German scientists went through and did a survey of different chemicals looking for those that had chemotherapeutic effects against bacteria. And they were using mice as their test models. They would infect the mice with bacteria, then inject them with various chemicals and see if it helped them recover faster. Now, one of the ones that became of high use was a chemical called Prontosil Red. It's actually a synthetic dye. When it was injected into mice, they were covered from various infections. Well, as it turns out, Prontosil Red contains a precursor of sulfa drugs. And we'll talk more about how sulfa drugs inhibit the action of bacteria, but this is one of our first commercially available chemotherapeutic agents. Sulfa drugs were used during World War II, and toward the end of World War II, we were able to isolate a type of penicillium mold that produced high quantities of penicillin, and that led to our ability to produce it commercially, and that also became of use to us. And so sulfa drugs, penicillin, they're the first of the antibiotics that we use today, and we've continued to find more and more products that are produced by microbes to inhibit the growth of other microbes. It's a battle down there in the soil, and if you can produce something that slows down or kills your competitors, that's all to the good of you and your descendants. But we're making use of it to inhibit the growth of bacteria inside of humans, or to kill bacteria inside of humans so that our immune system can get caught up and wipe out the invaders. Okay? Antibiotics are an aid to the immune system. Now let's talk about two different terms, broad spectrum versus narrow spectrum antimicrobials. Narrow spectrum antimicrobials are specific against just one group of bacteria. The first penicillin G, that we isolated from the Penicillium mold, is narrow spectrum. It only affects gram-positive bacteria. It can affect gram-negative bacteria, but you need much higher quantities. Now, broad spectrum antimicrobials affect more than one group of bacteria or microbes. So, for example, tetracycline, we'll be talking about that one more later, affects both gram-negative and gram-positive bacteria. So that would be an example of a broad-spectrum antimicrobial. Now, you have to balance what the benefits of a broad-spectrum antibiotic versus a narrow-spectrum one if you don't know what's causing the illness. It's nice to be able to use a broad-spectrum antibiotic because you know it's going to take care of whatever, and you don't have to wait for the results to come back from the lab before the patient starts feeling better. On the other hand, broad-spectrum antibiotics can lead to superinfections. That's where you decrease the numbers of your normal flora from the state of health to the point where one or two, usually one, type of bacterium takes over and increase in numbers to the point where it causes disease. Clostridium difficile is a classic example of this. Also, when women get vaginal yeast infections, oftentimes it's because they've been using a broad-spectrum antibiotic, and that wipes out the normal lactobacillus that are in the vaginal tract, and this causes Candida albicans, a yeast that is normally present in small amounts in the vagina, to increase in numbers to the point where we have disease. So you can see the benefits and the hazards of using a broad-spectrum antibiotic. Now, if you use a narrow-spectrum antibiotic, you have less risk of superinfection. But generally speaking, you have to know what's causing the illness. So it takes time to culture in the lab a sample and find out what's causing the illness. You have a delay in treating the patient feels bad for longer and is possibly spreading the illness. So those are the benefits and the hazards of using a narrow spectrum antibiotic. So you have to balance these in a clinical situation. Before we start talking about specific antibiotics and antimicrobials and their actions, let's talk about antimicrobial action in general. There are bactericidal products, and these are drugs that actually kill the bacteria. Remember, cidal means that it's killing. Then you have bacteriostatic drugs. These don't actually kill the bacteria, but they stop them from growing. And if they're not growing, then your immune system has an easier time coming in and killing them. So within the body, you have these two different actions, and it just depends upon which is best suited to the clinical situation. Now, as far as antimicrobial action, whether it be bacteriostatic or bactericidal, you have good targets for these antibiotics, and you have not so good targets. So let's talk about what some of the good targets would be. Anything that's unique to the pathogen is going to be a good target, because if the antimicrobial can target something that's unique, it's going to leave you alone. Anything that's a shared process or structure. In other words, if it's shooting our cells as well as the bacteria, in war we call this collateral damage, and you don't want that within the body. Actually, in war you don't want that either. So that's why we generally don't go after shared processes like glycolysis. We don't want something that shuts down glycolysis because that'll shut down our cells as well as the bacterial cells. That would be a not so good target. So we're going to redline that one. Now, we can attack unique parts of shared processes. Over time and with accumulated mutations, some parts of shared processes are unique to the pathogen, and ours is just different enough that the antimicrobial doesn't affect those enzymes as much as it does the enzymes of the pathogen. But those are harder to come by than antimicrobials that actually attack enzymes that are unique to the pathogen. Okay, cell wall synthesis, that's unique to pathogens. Animals, such as yourself, your cells do not have cell walls, so we can attack cell wall synthesis and it won't bother us at all. Protein synthesis. Now you're probably thinking, now how is that a unique process to the pathogen? We make protein ourselves. Well, remember back to when we talked about translation and also the parts of the cell, and I said you need to remember the sizes of ribosomes. Remember that? Think back. What's the size of the ribosome of prokaryotes? 70S. What's the size of the ribosomes of eukaryotes? 80S. They're different sizes, they're different shapes. So an antimicrobial that inhibits the actions of 70S ribosomes is going to leave 80S ribosomes alone. Now, as we go along, we'll talk about this. There are some times when drugs that affect 70S ribosomal function also affect the functioning of your mitochondria. Why? Because they have 70S ribosomes. But they're different enough that usually these drugs will leave your mitochondria alone. There are some that don't. You have to watch those. Be very careful with them. There are some differences in metabolism between some prokaryotes and us, and we'll be talking about that as we go further along. But one of them is the synthesis of folic acid. Now, folic acid is a B vitamin that we, as humans, cannot make ourselves, but bacteria can make them. So we can attack the process of folic acid synthesis, shut that down, and it shuts down the growth of protozoa and prokaryotes. DNA synthesis. If you can inhibit synthesis of certain of DNA, then you can shut down the growth of a certain pathogen. That's harder to do. Okay, cell membrane. All cells have a cell membrane, so this is a shared structure. But there are molecules within cell membranes of the different classes of microbes that we can attack, okay, that we don't have. One of these is types of antifungals that attack fungal membranes and leaves ours alone. Okay, and also we can attack receptors, enzymes, speed of metabolism, structural molecules, etc. Generally, we're going after the unique parts. Now, when we were talking in the previous chapter about anti-HIV drugs, what did we attack? Enzymes that were unique to HIV, unique to retroviral viruses. And so we can attack those specific enzymes and leave the patient relatively unaffected. Okay, so as we go along and we talk about specific antimicrobials, we're going to be grouping them into these various categories. So keep this in mind as we talk about the drugs going forward. Now, the common drugs that we're going to talk about those that inhibit cell wall synthesis and protein synthesis. Those are the bulk of the drugs that you're going to be familiar with. Like I said, good targets. We have a lot of drugs that attack those. Then we're going to talk about those that attack parts of the plasma membrane. We're going to talk about those that inhibit nucleic acid synthesis, synthesis of essential metabolites, specifically folic acid, antifungals, and antivirals. Let's begin with those drugs that attack cell wall synthesis. Penicillin is a part of a group of drugs called beta-lactams because they all have this beta-lactam ring in the center. We call this a part of the nucleus of the penicillin. Cephalosporin is also a beta-lactam. And this is the part that is needed to be active in inhibiting cell wall synthesis. Gram-positives are more sensitive to this. Now, later on when we talk about resistance, we'll talk about how penicillinases can go in and cleave this beta-lactam ring, inactivating the penicillin. Now, how does penicillin and the other beta-lactams inhibit cell wall synthesis? Well, they prevent cross-linking of the peptidoglycan units with the tetrapeptides. If you don't remember this, make sure you go back to the cell parts. Vancomycin also inhibits cell wall synthesis, but it's not a beta-lactam. If you look at that, you can see why that would inhibit cell wall synthesis. It's a big, scary molecule, and it prevents cell wall synthesis. And we have a tendency to use vancomycin as a drug of last resort. It has a fairly high level of toxicity relative to therapeutic ability. Now the last one, azonazid, and that one inhibits mycolic acid synthesis. So what organisms is this specific against? That's right, it's specific against acid-fast bacteria. Now let's talk about some of the more common drugs that inhibit protein synthesis. Let's talk about chloramphenicol first. Chloramphenicol changes the shape of the 50S subunit of the bacterial ribosome, that's the large subunit, and prevents it from functioning and attaching amino acids to each other to form proteins. Now, aminoglycosides, examples of that are streptomycin and gentamicin. They kind of do the opposite thing. They attach to the small subunit, the 30S ribosomal subunit, and prevent it from attaching to the 50S ribosomal unit, so that the ribosome can't clamp over the mRNA, and that inhibits protein synthesis. And lastly, tetracyclines. They inhibit binding of the tRNA of the ribosome, and this also inhibits protein synthesis. Now, because they attack 70S ribosomes, all of these drugs are broad spectrum. They have a tendency to be effective against both gram-positives and gram-negative, and to a certain extent against acid-fast. Not so much against acid-fast, simply because acid-fast bacteria grows so slowly. Antibiotics that attack cell wall synthesis, on the other hand, have a tendency to be more effective against gram-positives. At least the penicillins are. Now let's talk about drugs that attack the plasma membrane. Isoniazid, you may remember that from this cell wall synthesis. Isoniazid specifically attacks fatty acid synthesis of acid-fast bacteria, and so it's effective against mycolic acid, and it's also somewhat effective against formation of the plasma membrane of acid-fast bacteria. Now, polymyxin B also inhibits fatty acid synthesis, but it's mainly effective against gram-negative bacteria, and for a long time it was the only thing that we had that would kill Pseudomonas bacteria. Pseudomonas bacteria are opportunistic pathogens. They're common soil bacteria. Generally they're not a problem unless you have a burn. Sometimes it'll get into the ears and cause problems, and once it gets in causing an infection, it's really hard to get out. Nowadays, polymyxin B is found in antibiotic ointments that you can purchase without a prescription. Now on to nucleic acid synthesis inhibitors. The first one is the rifamycin family, and this is effective against acid-fast bacteria, so they have a tendency to combine this drug with azonazid. And an interesting side effect of this one is that it turns your fluids orange-red. So it's usually your urine, but it can show up in sweat and tears. Now with your quinolones and your fluoroquinolones, these attack bacterial DNA gyrase. Now gyrases are basically a class of helicases. So what do helicases do? That's right, they unwind the bacterial chromosome and unzip it at the replication fork so that the bacterial chromosome can be replicated prior to the cell dividing. Well, these inhibit that, and so the cell can't divide. And your immune system has an easier time taking it on. Ciprofloxacin is the most common one, at least the one that you may have heard of. It's generally known as Cipro, which is its brand name. And it's one of the things that was given to the folks that were infected with anthrax when the spores were being sent through the mail in 2001. Now let's talk about sulfa drugs again and talk about how they inhibit the growth of bacteria and some protozoa. We as humans cannot make our own folic acid, but some bacteria and most protozoa can. Let's talk about the process of making folic acid and why folic acid is important. Okay, dihydrofolic acid is composed of 3 parts. That gets converted into tetrahydrofolic acid, which is a precursor of DNA and RNA. So if you don't have folic acid, then you can't make DNA and RNA, so you can't divide and grow. And also, if you can't make mRNAs and you can't make tRNAs, you can't make proteins. So that inhibits the growth of cells. So let's talk about how bacteria that have the right enzymes can make folic acid. So one of the precursors, PABA, goes into this section of dihydrofolic acid. Now, sulfa drugs look like PABA, which goes into this portion of dihydrofolic acid. So the enzyme that takes these 3 components and smushes them together recognizes this portion as being PABA, and it puts it into the active site, and because of this extra part of the molecule, it doesn't fit just right. So the enzyme has to spit all 3 out and try again. And this is called, that's right, competitive inhibition. So sulfa drugs engage in competitive inhibition of the enzyme that creates dihydrofolic acid. Now, there's another drug, tetramethoprim, that looks like it's a structural analog of this portion of dihydrofolic acid. So if the enzyme recognizes this portion, it goes in, it doesn't fit. Once again, it's got to spit it out. Both of these drugs inhibit the formation of dihydrofolic acid, which inhibits and kills bacteria. Now, I've mentioned that sulfa drugs can inhibit protozoa. That's because they have a cell wall, and they can't transport folic acid inside past that cell wall. They don't have the proper transporter protein. So they have to make their own folic acid. So when they're exposed to sulfa drugs and they can't make their own folic acid, then they die. So that's as far as I'm going to go with the antiprotozoal drugs. There's an interesting section in the book about antimalarial drugs, but now we're going to move on to other eukaryotes, specifically fungus. Now, we don't have a whole lot of good drugs that are active against fungus because they're eukaryotes like us. It's difficult to attack them. The only one that I want you to remember is amphotericin B. Amphotericin B is one of the most commonly used antifungals, and it has a fairly high toxicity rate, so you're only going to be prescribing this to patients who are in the hospital or who can be closely monitored. Now on to antivirals. Now, we talked about antivirals in the last chapter when we talked about HIV, and those drugs are unique enzyme inhibitors. I'm not going to go over them again. Make sure that you understand them from the last chapter. Interferons we've talked about before in the chapter on innate immunity. Make sure you understand those and how they inhibit viruses. Now let's talk about nucleoside and nucleotide analogs. This is another situation of competitive inhibition. This is where we have, okay, here's a guanine, okay, that's the G in DNA and RNA. And acyclovir, which is one of the more common nucleotide analogs, looks like guanine. It has a nitrogenous base. But, oh, look down here, we don't have the whole rest of the sugar. And if you don't have the sugar with a phosphate, you can't connect them together to make that sugar-phosphate backbone. So it shuts off DNA synthesis when it gets incorporated. Okay. Acyclovir has been highly effective in use against various herpes viruses. It also has a tendency to be used for HIV positive patients. It does work somewhat against retroviruses, but it's mainly effective against various herpes viruses that come out of latency, come out of hiding when the immune system drops in patients with active AIDS. It also gets used for people that have severe herpes infections. For example, if you have a patient that has a really bad case of shingles, then we can give them acyclovir and help get that under control. Well, that's it for part 1. In part 2, we're going to go over the ways that we test the effectiveness of various antimicrobials, and also how bacteria develop resistance to antimicrobials.